So, good morning. To a topic today, which am I audible actually?
Yes, sir.
Okay. So, the topic today, which I will be taking up, is on mitochondria. So, without wasting time, I will try to share the screen. So, what exactly is a mitochondria? Mitochondria is actually the powerhouse of the cell. Mitochondria is the powerhouse of the cell. That means it provides energy for the cell to do all its activities. It is one of the major source of energy. They were first seen by Goliker in 1850 in muscles, and they called them as sarcosomes. Fleming in 1882 described these organelles as philia. Altman in 1890 observed these and named them as bioblasts. Benda in 1898 stained these organelles with crystal violet. It's a dye, and called them as mitochondria. Then Michaelis in 1900 used Janus green B as a vital stain to observe mitochondria in living cells. Several important Nobel Prize have been won. Especially the most notable was that of Peter Michel. You had the Kemi osmotic theory, got the Nobel Prize. He was also a faculty in the university where I did my PhD in Frankfurt. He was essentially from Frankfurt, and then he studies at by University Biocenter Max Planck Institute in Frankfurt. And the shape of the mitochondria, it is highly variable. It can be short rod shape to elongate filamentous form. It can be rod shape like this. It can be like this. It can be also like this filament. Its size is variable. It measures from 0.5 to 2 micrometer in diameter. They have an average length of 3 to 4 micrometer. The number of mitochondria varies from one cell type to the other. Obviously, it depends on the cell. So cells which have got which need lot of energy, they have lots of mitochondria. Less energy, less mitochondria. They are not found in prokaryotes, so they are exclusively eukaryotic. So if you look at the structure of the mitochondria, Let me allow me to draw this on the whiteboard. If you draw the structure of a mitochondria, you have an outer membrane. This is the outer membrane. Then you have an inner membrane which is folded into finger-like projections. It is folded into finger-like projections. This is the inner membrane into and these are finger-like projections and they are known as cristae. Purpose of it is to increase surface area. Because on these membranes, there are enzyme complex on the inner membrane. There are enzyme complexes throughout the membrane, and they are basically the enzyme complexes of the electron transport chain. This is the mitochondrial matrix. This contains all the enzymes of a step known as Krebs cycle. And this is the intermembrane space. This is the intermembrane space. A characteristic feature of this intermembrane space is concentration of protons. H plus is protons is high in the intermembrane space. And the mitochondrial with several enzymes of biosynthetic pathway are present here. And don't forget the electron transport chain. Complexes of the electron transport chain. We will talk about it in more details a bit later. As you can see, and characteristic feature is mitochondria have their own DNA. Have their own DNA. Own ribosomes. So these folded regions are known as cristae. And this dark brown color, it is the intermembrane space. The central region is the mitochondrial matrix. And ATP synthase, it's a component of the electron transport chain. Electron transport chain, or ETC. This is an electron micrograph. This picture was taken from electron microscope. You see, you see these crystal-like structures present, folded like this on the top. Okay. Now, if you look at the ultrastructure, again, I'll repeat. A mitochondria is enclosed by a double membrane envelope composed of lipid and protein. The two membranes are separated by a narrow fluid filled space called the outer compartment. The outer membrane is smooth. It is more permeable to small molecules and it contains some enzymes but is poorer in proteins. The inner membrane surrounds a central cavity or matrix, inner compartment. The folds of the inner wall of the mitochondria are known as cristae. So the outer membrane is smooth. It is more permeable. The inner membrane is not that permeable. It is permeable to small molecules and contains some enzymes but is poorer in proteins. এক সেকেন্ড ধরো সুন্দর। So the inner membrane, like I told, the most important part, the inner membrane, it is the site of electron transport chain across which the proton pump occurs. I will explain. And contains the enzyme ATP synthase. Inner membrane is highly folded. It increases the surface area on which the electron transport chain reactions takes place. Electron transport chain, main function of electron transport chain is synthesis of ATP. Main function is synthesis of ATP. Okay, mitochondrial. So it is compartmentalized. That means the chambers into numerous cristae which expand the surface area of the inner mitochondrial membrane. And this enhances the ability to generate ATP, or the energy currency of the cell. In a typical liver mitochondria, the surface area, including cristae, is about 5 times that of the outer membrane. So, inner membrane, surface area, keep in mind, 5 times more than outer membrane. Mitochondria, muscle cells, it's very high. Christie in muscle cells, mitochondria is higher than Christie in liver cell mitochondria. So this is one aspect of it. Now let us talk about the outer membrane. The outer membrane, as you know, it encloses the whole organelle. This brown, light brown colored structure. It has a protein to phospholipid ratio. Phospholipids are lipid plus phosphate. So basically, in a plasma membrane, I've seen you have a hydrophilic head, and a hydrophobic tail. It's like similar to that of the plasma membrane. It contains, as usual, several integral proteins which span the membranes, and they are known as porins. So remember, porins. They are integral membrane proteins. Integral membrane proteins. Basically, they act as channel. They act as channels. What does a channel do? Say a barrier is there. This protein, it allows entry. So this is the channel protein. It is permeable to molecules of 5,000 Daltons or less. Larger molecule to enter inside, they need active transport. You will find it on my note on in the plasma membrane, in my slides of plasma membrane, active transports. So double membrane creates two spaces: matrix, large internal space of matrix, and a space, intermembrane space between the membranes. So this is we talk about intermembrane space. Again, I just remind you that concentration of hydrogen ions or protons, they are all known as protons, is higher than that of mitochondrial matrix. This is very important for electron transport chain functioning, and most essential for ATP synthesis. So some functions of the matrix are being given. It contains a highly concentrated mixture of hundreds of enzymes, including those required for oxidation of pyruvate and fatty acid and for the citric acid cycle. Fatty acid synthesis, citric acid cycle, all these things, it contains all the enzymes, beta oxidation enzymes, the fatty acid synthesis. These are all biochemical pathways which take. You will read about these pathways in biochemistry. The matrix also contains several identical copies of mitochondrial DNA genome, special mitochondrial ribosomes, transfer RNAs, and various enzymes required for the expression of mitochondrial genes. tRNAs, as you know, are very important for protein synthesis. Protein synthesis, and they are also adapters. They attach they connect the messenger RNA to the protein translating machinery. So RNA to protein synthesis, the adapter are the tRNAs. So now we talk about the inner membrane. Inner membrane in the red color, it is folded into numerous cristae which increase its total surface area. It contains proteins with three types of functions. One, that those carry out oxidation reactions of the electron transport chain. Electron transport chain has got a series of oxidation. That is, what is oxidation and what is reduction? So gain of electrons is reduction, loss of electrons, oxidation. So all these oxidation reactions take place there. The ATP synthase that makes ATP in the matrix, it's also present here. ATP synthase structure is, it's a knob shaped structure. It's like a dynamo. So it's present on this membrane. This is the high concentration of H plus ions over here in the intermembrane space. So, and there is low concentration of H plus protons inside. So they enter through this high concentration to low concentration, and it acts like a dynamo. And that dynamo, what it does, it rotates and it generates energy needed for ATP synthesis. This is the fantastic function of this particular ATP synthase. And it has also got transport proteins that allow passage of metabolites into and outside the matrix. So all components of the citric acid are metabolites like lactate, malate, all these things. There are transporters which take them inside. It is a large channel forming protein called porine. The outer membrane is permeable to all molecules 5,000 daltons or less. Other proteins in this membranes include enzymes involved in mitochondrial lipid synthesis. So enzymes needed in fat synthesis in the mitochondria that convert lipid substances into forms that are subsequently metabolized in the matrix. And in the intermembrane space contains uh, enzymes that use ATP passing out of the matrix to phosphorylate other nucleotides, and it has got high concentration of H plus ions. So chemical composition, 70% lipids, 70% proteins, 25-30% lipids. Mitochondria also contain 0.5% RNA and other traces of DNA. Mitochondrial DNA comprises of 1% of total cell DNA. And mitochondria contain enzymes for oxidation, phosphorylation, and electron transfer. So function, what it does, it produces most of the ATP of the cell. Krebs cycle also takes place inside, that also produces ATP. NADH is also an electron carrier which is present, very important compound. It donates the electron to the electron transport chain and becomes oxidized to NAD+. This we will be studying much more details right now. Keep this line in mind. Electron transfer promotes proton pump and ATP synthesis in a process known as oxidative phosphorylation. I will talk about it just now. After this, I finish this. So they contain their own copies of DNA and RNA along with transcription and translation system. Transcription is the synthesis of RNA and translation is the synthesis of proteins with the help of ribosomes. They are able to regenerate themselves without the whole cell undergoing division. So the mitochondria can divide inside the cell itself. They don't need the cell for this. Shape and size dependent on what the cell's function is. So this is the first part. The next part which we will go is the Kemi osmotic hypothesis. Could some of you confirm if you are able to see this picture?
Yes, sir.
Okay. So you see this is the outer membrane. Outer membrane. This is the intermembrane space of the mitochondria. And this is the inner membrane folded as cristae. On top of the inner membrane, there are electron transport chain complexes. This is complex 1. Then you have the complex 2. You have the complex 3. And you have the complex 4 present over here. Now, when Krebs cycle takes place, Krebs cycle, glycolysis, they generate a compound known as a compound with high reducing potential. That is, they've got an energy. They can donate electrons and hydrogen very superbly. NADH and FADH2. NADH breaks up into NADH plus and H plus. It breaks up into NAD plus plus H2 plus 2 electrons. So this NADH is produced from Krebs cycle, glycolysis, fatty acid synthesis, beta oxidation. These are the main processes which produce this NADH and FADH2. So that is present inside the mitochondria. This breaks. What it does in the process, the hydrogen ions, one from here, and 2, it pumps out 4 hydrogen ions outside. So in the intermembrane space, so the concentration of H plus here, it increases, or the protons plus charge increases. This NADH then releases electrons here to the complex 1. This has got high energy. This is a high energy electrons. So this high energy electrons, it loses some of its energy and pumps hydrogen ions outside, increasing the hydrogen ion concentration in this region. In the intermembrane space region. Where is the energy for pumping out H plus ions? The energy comes from the high energy electron. It releases some of its energy to pump H plus ions. Then the H plus goes to a molecule known as quinol. Ubiquinone, you can call. It goes to this Q molecule. Similarly, in the complex 2, the same function takes place and this gets reduced and electron is transferred into Q. And now they go to all of the electrons, they go to the third complex. Here more energy is given and more H plus is pumped out. Again, it goes to complex 4. You don't need to study all the complexes, function of all the complexes now, just a brief overview. And what you find over here is in the complex 4, you have more hydrogen ions being pumped out. Finally, water is produced here and oxygen is needed. And then you have the fifth step. This is your famous ATP synthase. Fine. And the high H plus concentration over here, and you have a low H plus concentration inside the matrix. This, from a higher concentration, it tries to go to a lower concentration. So this H plus enters inside. And when it enters this unit, it rotates. It rotates like a dynamo, cycle dynamo. It keeps on rotating. And this rotation generates energy. And that energy is utilized for making ATP from ADP and inorganic phosphate in the cell. And this ATP is the energy currency of the cell. The cell needs ATP as an energy. This theory is known as it was postulated by Peter Michel in 1961. It's known as the Kemi osmotic theory or Kemi osmotic hypothesis. So what happens over here is pumping out of H plus into intermembrane space what it results, it generates a proton motive force. This H plus is a proton. And what it does is that when this H plus goes inside, there is this ATP synthase enzyme, and that turns like a dynamo. When H plus from higher concentration, like water is flowing and a turbine is being moved, and this causes generation of electricity or energy. So energy is produced, and this energy is utilized for making ADP plus PI into ATP, which is a high energy in a compound. This is the chemiosmotic theory. Now let us talk a bit about mitochondrial DNA. This is the mitochondrial DNA complexity in different organisms. You remember only these three. This data, just remember only these three. I would say just remember, if you can remember any mouse and human or only the human, that's fine. Just remember one thing only. Don't have to understand, remember all of them. So what are the properties of mitochondrial DNA? So the mitochondrial DNA from a wide variety of species shows great similarities and has consistent biond density. So they are very similar to each other. Melting temperature of mitochondrial DNA differs from nuclear DNA. So what is melting temperature? Let me draw this on the whiteboard. So DNA is supercoiled. It's in a double helical strand. This, when there is a temperature, this strand becomes separate, and that is known as the melting temperature. single-stranded DNA. So this is the melting temperature. So the melting temperature of mitochondrial DNA is different from that of nuclear DNA. Where is nuclear DNA present? In the nucleus, as we have studied in the last class. The mitochondrial DNA is shorter than the nuclear DNA. So they can independently DNA replication takes place. This is the difference between the genetic code of mitochondria and the universal genetic code from the nucleus. There are some differences. Just briefly read those things. And you need to practice the structure of the yeast mitochondrial genome. What are the different genes and all present over here? Just practice this one. So with this, I actually finish today's class. I would be happy if you have some questions.
স্যার আপনি যে বললেন এটিপি সিনথেসটা ডায়নামোর মতন ঘুরে, ওটা কি এটিপি গেলেই ঘুরে নাকি ওটা নেচারটাই মানে ঘুরে ও জেনারেট করে এনার্জি?
দেখাচ্ছি কিভাবে হয় ওটা। দেখি যদি কোনো ইউটিউব ভিডিও পাওয়া যায়।
My job is to make college easier because students have a lot.
দেখি এটা শেয়ার করি আগে।
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized. The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
So, বুঝতে পারলে এটা? Were you able to understand?
ইয়েস স্যার।
Okay, okay.
আর স্যার আরেকটা বলবো, আপনি যে বললেন না এনজাইমটা পাওয়া যাচ্ছে ফ্যাটি অ্যাসিড সিনথেসিসের জন্য ম্যাট্রিক্সেও আর আউটার মেমব্রেনেও। স্যার মানে তাহলে সিনথেসিসটা মানে ম্যাট্রিক্সে হয় নাকি আউটার মেমব্রেনে হয় ফ্যাটি অ্যাসিডের?
ফ্যাটি অ্যাসিডের সিনথেসিসটা ভেতরে ম্যাট্রিক্সে।
ওকে স্যার।
ম্যাট্রিক্সে হয়। ঠিক আছে, রাখছি তাহলে আজকে।
ওকে স্যার।
ওকে স্যার।